this wonderful webinar. I'm gonna share my screen, okay? So let me just 1 second and open my PowerPoint. Can everybody see my screen now? It's all good? Excellent. So, okay, hello everyone. Michael, first of all, thank you for your work on this report on the new species described last year about continental fishes. And I'm glad to be part of this seminar. My name is Jairo Arroyave, and I am a researcher and professor at the Institute of Biology of the National University in Mexico City, UNAM. So today I'll be talking about the discovery and description of Easternum berlini, a fish that doesn't live in the water. And the idea is to give you some of the context of the discovery, but before I'll be talking about generalities of this group of weird fishes and then what we did exactly to describe this new fish. So Ophisternum berlini is a member of a family known as Symbrachidae. They are commonly known as swamp eels. So just from the name, you know that they live in swampy areas, or they can live part of their lives in swampy areas. Despite the name eels, they're not really true eels. They are a very distantly phylogenetic group of fishes. They're not true eels who are in the order Anguilliformis. Symbrachidae are mostly freshwater. They can be sometimes estuarine. They're really never marine. As I was saying before, they include a lot of species that inhabit swamps and have the potential to breathe air. However, they're normally associated to, like these swampy areas are associated to more like permanent bodies of water. They're taxonomically challenging because of their conserved morphology. As you see, I don't know if you can see my pointer here, but like you see the body shape, the external morphology is very simple. They lack a lot of characters that we taxonomists use to differentiate a species based on external morphology. They don't lack they don't have fins, paired or median fins, and their morphologies are pretty conserved. In the case of this family, we have a total of 28 species, including the one that we just described last year. And these species are distributed in 7 genera. I've listed all these genera here, and the number of species in each genera and their distribution. As you can see, the family is widely distributed worldwide. Here in the map, we can see where they are. I mean, there's certain gaps in the distribution, but they're like in pretty much almost every continent. And the genera that I think are more important for my presentation today are, of course, the genus of the species we described, Ophisternum, and Symbrancus, which is another genus of Symbrachidae in the neotropics. Ophisternum is perhaps the most widely distributed genus in the family. With the species we described, there are now 7 species valid, and they are basically in most of the distribution for the family. And in Middle America, there's most of its diversity is focused or concentrated in Middle America. So difference, as I was saying before, there's different distribution patterns between these two genera that are present in the New World. Ophisternum is way beyond the New World, and we have species in West and Central Africa, South and Southeast Asia, and Australia. And Symbrancus, who is represented here by the stripes. The area in stripes is distributed in Central and South America. So according to this map, supposedly there's some area of symmetry, but I'm not quite sure if this is still considered, because my understanding of distribution in Ophisternum is that it's basically restricted to Middle America, Southern Mexico, Belize, Guatemala, sort of and with the new species, Costa Rica. We'll talk about that later. So this is how a Symbrancus looks. This is the type species, Symbrancus marmoratus. You see what I was saying before, that is a species with a very conserved external morphology, like very simple, paired and medial fins, and this is based on some historical illustrations and some photographs from a more recent publication. And Ophisternum is externally very similar, very similar. These are just some images from the original description, which dates back to the 19th century. And with the first species described actually or like they were like taxonomic changes, because at some point it was considered as a member of Symbracus. The taxonomy is very convoluted, but anyway, how to distinguish between these two genera of neotropical swamp eels? Externally, normally we have to look at the brachiostegal membrane. These fishes of the family Symbrachidae have this type of brachiostegal membrane. This is the membrane that covers basically the gills, and sometimes is united and free from this ventral part of the fish. And depending on basically the arrangement of that union of the membranes from each side, we can have different shapes. And supposedly, our understanding is that in Symbrancus, the shape is more circular. They form kind of like a little pore. That's the opening of the membrane. Whereas in the genus of Ophisternum, it tends to be more like a slit, which can be curved or can be less curved, can be wide or can be a little shorter, as in the new species you will see. Here's just another slide showing you some of the differences between Ophisternum here, and this would be the opening of that membrane in Symbrancus. So these characters sometimes, I mean, here is very obvious, like we see the slit is very large, the pore is very small, but still sometimes it can be a little tricky to distinguish based on that external morphology. Internally, however, there's a more straightforward way to distinguish between these two genera. And the structure has to do with the support that most fishes or teleost fishes, bony fishes have for the pectoral fin. And this internal bony support is known as the pectoral girdle, which is all this structure here that we see. We have like a secondary part, all this arch of different bones, and there's another section here where the pectoral fin will attach. And even though I've mentioned that Symbrachids have no fins, right? They still, interestingly, they still retain the bony support, the internal pectoral girdle. And when we look at the structure of that pectoral girdle, we see a huge, like a very obvious difference between Ophisternum and Symbrancus. Ophisternum has like I would say like a more normal pectoral girdle. This would be the disposition of the different bones. They are basically attached to the skull by the posttemporal here. And the cleithrum is basically at like joining the axial skeleton at the third vertebra. Whereas in Symbrancus, we see a displacement, posterior displacement towards the back of the fish, all the way to the fifth vertebra, and the posttemporal is highly reduced to a rod, as you can see here, compared to this piece, and detached from the rest of the girdle. So if we look at the internal anatomy, like the osteology, we can easily distinguish these two genera. Now, neotropical Ophisternum, as I was saying, is like where most Ophisternum are in this neotropical region. Prior to our description, our discovery, we knew about two species of Ophisternum. One is present in southern Mexico, Belize, Guatemala. This species is known as Ophisternum enigmaticum. Here are some pictures of one that I actually collected like a few months ago. And the other one, well, this one is, as you can see, is widely distributed, is mostly present in, like lakes, rivers. It has like a lot of epicontinental water systems that it can be found. This one is more special because Ophisternum infernale is endemic and restricted to these subterranean waters of the Yucatan Peninsula aquifer. Basically submerged caves that are present in this beautiful system. This is how those caves look in this video. Actually, I'm here diving, looking for Ophisternum infernale, as well as the other endemic species from the system, the other cave endemic, called Typhias pearcii. It's a blind brochure. So, two species of Zimbrancus have been described in the 21st century. And these are the two species of Zimbrancus that have kind of recently been described, whereas for Ophisternum, there hadn't been any descriptions of new species in the past 50 years. The last one was described in 76 in this seminal work by Rosen and Greenwood. So you can see how much time had passed since any Ophisternum had been described. So now let's talk about the discovery of our own species. So this species, as you may know, was described from Costa Rica. Is present in, as far as we know, is only found in this small area in a nature's reserve in the Atlantic slope of Costa Rica. Here is basically where we found it. The reserve is called Las Brisas. This is, maybe here you can see a river going through this area, and that's this river pictured in this beautiful photo. This is the Destierro River. And perhaps looking at this satellite image, you can see that here's the river, and there's an area of like forest that is in pretty good shape, but surrounded by a lot of agriculture and sections with clearly the forest has been removed and replaced by a lot of agricultural land. So this is important to mention here because this little area of reserve that is protected is where we found the fish. So we don't know if it's anywhere else outside this area. Probably not, and the importance of having this area conserved given the high impact of human activities around the area. So that's the location of the where the species lives. This is the habitat. This species was not found in water. This species was basically found in a swampy area, like buried in the soil in this reserve. Here in this map, you see the Destierro River, and the first specimens are located from this section. In 2021, there are specimens, 2022 and 2023. I'll talk more about this shortly. And this is how the fish looks. So it does look like kind of like a worm at first sight. I think most people wouldn't think that this is a fish, also because it's very tiny. However, the owner of the reserve, Mr. Eric Berlin, was insightful enough to think that this was a really strange thing, that most likely was a fish, and therefore he reached out to Dr. Arturo Angulo at the University of Costa Rica to tell him about this weird discovery and if he could shed some light on the identity of this creature. And so Eric sent Arturo around 2021 basically a block of mud where the fishes were found. And Arturo in the lab basically had to dig them out. And this is the video that he got that time, where we see a couple of specimens. And yeah, clearly there were fishes, and Arturo right away was able to tell that they were most likely Zimbrancid eels. However, when he started looking at the diversity of Zimbrancid eels in the neotropics, he couldn't find really anything that looked like that was previously described, that we knew about. It was not the species Zimbrancus marmoratus, which is like a Zimbrancid fish that is known to occur in this area, in the rivers and like lakes of this region, it was clearly different from Zimbrancus marmoratus. Uh, it was actually very similar to a species of the genus of Sternum that lives in the other side of the world, all the way in Australia. Um, and that's when Arturo thought that maybe this could be a specimen of Sternum. However, it was really weird that the most similar thing was so far away. Uh, Another very similar species, more, like in proximity to Costa Rica, not as far as Australia, is a species that is cave dwelling, cave adapted, troglomorphic from the Yucatan Peninsula aquifer of Eastern Infernali that I was talking about earlier. And we recently published a study precisely on that species and some of the evolutionary history based on mitochondrial genomic data. And I think Arturo got a hold of it. Oh, sorry, my dog is doing. Sorry, give me 1 second. Sorry about that. I'm back. So, so Arturo got to see this work and the similarity with the fish that he found, that because of some similarities, he reached out to me. And that's when basically we started to collaborate on this project, right? And part of that collaboration involved getting to the field and acquiring more specimens. So that was the plan. I told Arturo, we need more specimens because if we want to describe these, if this is really a new species, we need a larger number of specimens and ideally some fresh tissue samples so that we can do genetics work, right? So with this in mind, the specimens that he received first were from 2021. Then he actually went in 2022 to get some more specimens. And this is after that when we got in touch and we planned the 2023 expedition. And basically this is the trip that happened. We stayed obviously in the reserve. Eric Berlin, the owner of the reserves, is highly committed to conservation, is highly committed to the study of the biodiversity in his reserve. And he was very excited to have us. We stayed in this beautiful cabin inside the reserve. And we, in addition to looking for Ophisternum, we did also some surveys of other fishes in the area. We got lucky. We found 2 additional specimens for a total of 7 in the whole series from these 3 different years. This is actually me in the reserve digging out some fishes that we had found, and this is how they look, live in an aquarium after pulling them out of the mud. And we also found some Zimbrancus marmoratus. These are the Zimbrancus marmoratus that we found in that expedition. So we also had these as comparative material thinking for the description. So the next part was precisely that. Let's see if this is a new species. So this is time to do comparative biology, what I think is the most exciting part, in addition to fieldwork about our job. So for that, I invited 2 colleagues. Well, Arturo and I invited 2 colleagues to work with us. One of them is my former mentor, Melanie Stiasny, curator of fishes at the AMNH, and she is like an authority in fish anatomy. So it was great to have her here because we wanted to do some of that as part of this description, right? And my current postdoc, Fernando Mar, he also joined us to contribute with a lot of like this genetics component of the study because we didn't want to like restrict ourselves to the anatomical or morphological evidence, but also have genetic data to support the distinction of these species. So after the trip to Costa Rica and having some of these specimens, I spent some time in New York at the American Museum of Natural History, where I joined Melanie, and I brought some specimens from Zimbrancus and others from the new or like the undescribed species to do some anatomical studies. And for that, we used the microscopy and imaging facilities at the AMNH that have like a state-of-the-art micro CT scan where we can generate images of internal osteology, basically a bunch of X-rays in all of sorts of directions to create three-dimensional images of this skeleton, and instead of making dissections or clear and staining specimens, which is a more destructive, I guess, like approach to study internal anatomy. And also leveraging the amazing fish collection of the Department of Ichthyology at the museum, we could find comparative material to aid our description process. And speaking of comparative material, we use obviously the two species that are in the neotropics that are in Middle America of the genus Enigmaticum and Infernali that I talked about earlier. We also included as comparative material samples of Symbrancus marmoratus. The only Symbranchid species that we know is sympatric with the new species that lives in the same area. It doesn't necessarily live in that mud, but is found in waters in the area, right? In the river, in the lake or lakes. And then we use also other species of Sternum that is not neotropical, that is all the way in Australia, and as an outgroup further, like a more distantly related genus of Symbrachidials. So that was basically what we used as comparative material, and the type of data that we generated. As I was saying before, we did a lot of micro CT scanning to generate these three-dimensional images of the skeleton. And we also use some clear and stained specimens. So what are the systems that we focus on for these descriptions? We focus on the skull. We looked at different sections, or we look at the neurocranium, characters of the neurocranium, of the oral jaws, of the suspensoria, of the branchial basket, and the higher series. But in addition to the skull, we look at the vertebral column, and we looked at the pectoral girdle. This is particularly for the distinction between Ophisternum and Symbrancus. As I was saying before, that's the most straightforward to differentiate species of Ophisternum and Symbrancus. So, okay, we got all this data. It was time to start reconstructing these images and understanding the variation in osteology in these fishes, in all of this comparative material that we had. And that was really fun process to be able to find all these characters that were ultimately diagnostic for the species that would allow us to say with evidence and with support, yes, this is a different species and this is why. So in addition to the anatomical data, as I may have mentioned before, with my postdoc, we generated complete mitochondrial genomes, not only for the new species, but also for a species that didn't have that mitochondrial genome published, which is one of the closest relatives of Ophisternum enigmatum, one of the two other neotropical Ophisternum species. And we use published mitogenomes for the genus, including Infernali, that we had produced in previous years, Ophisternum candidum, the Australian species, Symbrancus marmoratus, and these are two other species from the family that are more distantly related. And we use these also in the analysis as the outgroup for phylogenetic analysis. So about the diagnosis, well, the first thing that we found is that indeed our fish is a member of Ophisternum. This is how the pectoral girdle looks like in Ophisternum berlini. This is Ophisternum enigmaticum, and this here is Infernali, Ophisternum infernali, and this is Symbrancus marmoratus. So it was clearly not a specimen of Symbrancus marmoratus, which is the other Symbrancid species found in the area. So with this, we already had enough evidence to place this fish in the genus Ophisternum. So in addition to that, we looked at the membrane, the branchiostegal membrane, and the opening, and we saw that we didn't have a pore, but we had a very strongly lunate slit. You see that is much smaller, in terms of the width of the fish in this section compared to Ophisternum infernale here, but it's not a pore like in Symbrancus marmoratus here. So that was another piece of evidence. This is in external morphology, suggesting that this fish was Ophisternum and not Symbrancus. Now, how do we differentiate these species from from the other Ophisternon in the neotropics and in the region, right, in Middle America. So I think the most obvious, the most conspicuous difference was the number of precaudal vertebrae. Even though all of these species have about the same number of total vertebrae, in fishes normally the vertebral column is divided in a precaudal and caudal vertebral region. And so basically all the precaudal vertebrae is where we have all the digestive system, like all the digestive tract. And then after the anus, then you have the caudal region where the caudal vertebrae are. So in all of these species, there are about 140 vertebrae, which is a lot, but it makes sense given the elongate shape of the fish. But in the new species, the precaudal vertebrae are very numerous, are 100 versus 65 or 75 in the other species. So this was like the first obvious difference between the new species and the other species in the region and in the genus. So in addition to this character, we found differences in dentition. Here we have the new species, and here the other two species. This is the premaxilla and the dentary. You can see in the premaxilla and the dentary, like very robust, large teeth compared to those in the other species, in Enigmatum or Infernale. They are like much smaller, not as robust, as large as what we have here. And in addition, we have also very large teeth in the palatine and ectopterygoid bones. Here, we don't have them as large as you can see in the other species, and also the number of rows in palatine teeth is only 1 for the new species, whereas we have 2 or 3 in Enigmaticum and Infernale, respectively. So that was another huge, very easy way to tell them apart, the dentition. When we compare the new species with Enigmatum, one obvious feature is the color, and this color and lack or eye reduction, I say, and lack of pigmentation, it has to do with this process known as troglomorphism, or this condition known as troglomorphism, which is the reduction of eyes and pigmentation. I mean, some people believe that this is an adaptation to these environments that lack light, right? So if you don't need eyes, if you don't need pigmentation, why have it, right? So in this case, when we compare the new species with Enigmaticum, the troglomorphism is a very strong difference and a way to differentiate them. However, Infernale is troglomorphic. So if we're comparing the two with this, troglomorphism is not really enough. But we looked at the internal anatomy and osteology, and we could see some major differences between the new species and the troglomorphic one from the Yucatan Peninsula aquifer. And first was like the lack of the neurocranial crest. This is present in Infernale, but is absent in the new species, right? Same with the size of this fifth ceratobranchial tooth plate and the dentition in these pharyngeal jaws is not as I mean it's kind of like more robust and larger than we have here in Infernale. The gill rakers also here are more elongate. They're like tiny, they're not a lot, but they're more elongate than what we have here. So there's also osteological differences between these species. And now looking at the genetics, we found like remarkable genetic differences. So that's another additional evidence for the distinction between the new species and the other two Middle American Ophisternon species. So we have these are just the percentages in genetic divergence in 3 different markers from the mitochondrial genome. The first one particularly is commonly used as the barcode, meaning like there tends to be a lot of conservation in that marker in members of the same species, but not so much between members of different species. So normally for freshwater fishes or for a lot of group of fishes, I think if we have like divergences of about 7% or more, sorry, I opened this. Sorry, 3% or more, we most likely are talking about different species. And here we had like a percentage of 15% compared to Enigmaticum, 14% compared to Infernali, 17% compared to the Australian Candidum, and almost 18% to the Simbranchus marmoratus. So in terms of genetic differentiation, we found like very large numbers, again, supporting the distinction of these taxon as different from what we knew before. And then we perform a phylogenetic analysis to kind of like place these species in the evolutionary tree of like Simbranchids. This is based on all the available mitochondrial genomes. And we found that the new species is the sister of the other species in the Neotropic that form a clade and are sister to each other, right? So this is one of the findings, and also our results support a previous finding that Simbranchus is actually nested within a larger clade of Ophisternon, where the Australian is here, and that renders the genus Ophisternon as paraphyletic. That means it's not a natural group. We have to work on at some point on the systematics of this group, because clearly there's something going on here with Simbranchus being nested within a clade of Ophisternon otherwise. We named the species Ophisternon berlini in honor of Mr. Eric Berlin, the owner of the reserve and discoverer of the population that we used to describe the species. And he's a great guy, a very strong supporter of conservation and scientific research in his reserve. He always has the doors open to biologists, to researchers who want to understand and contribute to conservation of the biodiversity in this area and in Costa Rica in general. So just to close my presentation, wanted to mention why is this claim that we believe Ophisternon berlini is effectively a fish that does not live in the water. So if you look at the title of our publication, which I may have not mentioned before, it was published last year in October in the journal Ichthyology and Herpetology, and we basically we highlight some conditions here that the new species is endogen, meaning it lives in the soil, is dwarf, and is troglomorphic, right? So we'll go a little deeper into these adjectives and why are we, highlighting them and using them in the title of our species description. So why do we think this species is dwarf, or what is the evidence that we have for dwarfism in this particular species? We found that adults that are very small, they might seem a little long, but they're really, really tiny. It's like this body plan of symbranchid eels. But like this length that I'm showing here is much smaller than what normally adults of congeners are. And we know they were adults because they were sexually mature. We actually found eggs we found like a female with eggs, so we knew it was already an adult because it was sexually mature. And we also have evidence from osteology suggesting that our specimens, even though they're tiny, they're adults. And this is related to the fact that they, here we have the new species. No, sorry, this is Ophisternon enigmaticum. This is the new species here. And compared to these, in these two, we have Ophisternon enigmaticum, CNDR Ophisternon, the skulls of Ophisternon enigmaticum in lateral and dorsal view. And you see that in the adult of enigmaticum, we have like evidence of a fully formed skull, where the sutures are kind of complete, are not open or still forming, as in these, like more like juvenile. And in juveniles also the roof of the skull is kind of domed. And this was basically the same size or around the same size as this guy that doesn't have this condition of a still developing skull. So we knew that at this size, at this small size, our new species is fully developed. And troglomorphic. Well, it's obvious, the eye reduction and the lack of pigmentation tells us that this species is troglomorphic. And troglomorphism is a condition in fishes that normally evolves in conditions of low light or, or complete darkness. which is mostly in caves, in submerged caves or in caves that are partially submerged with water. But the whole idea is that habitats that are completely dark. And the thing is that the soil is also dark. If we think of a species that lives inside the soil, well, there's no really sunlight reaching to these sections of the habitat, and therefore it's not that surprising to have a troglomorphic animal living in the soil. And actually, most of the diversity of symbranchid eels, or a large proportion of symbranchid eel diversity here we see, is troglomorphic. It's like 30-something%, if I recall correctly. Now, within the genus of Ophisternon, half of the diversity is troglomorphic. The new species, the Australian Candidum and the Mexican endemic Infernale, are troglomorphic. These two, however, live in caves, right? Berlina is the only one in the genus that lives in the soil. 100% of Rhactamictis diversity and other symbranchid eels from India, they are all troglomorphic. 100% of Typhus imbrancus in Western Africa, Western Central Africa, we have this diversity in the genus completely troglomorphic. And the only genus of Macrotrema, the only species, sorry, of Macrotrema in Southeast Asia is also troglomorphic. So there's a lot of troglomorphism in the family. However, berlini is only one of the 3 species that lives in the soil. And not only in symbranchid eels, but my understanding is that in all fishes, we only know 3 species that inhabit permanently the soil, and these are those. So they're members of the same family, and berlini is the new addition to the select list of endogenous fishes. So with this, I'd like to end my presentation, acknowledge my sources of funding, and also the Miami Herald, who published a little note about our discovery last year. And I guess with this, I'll finish. I guess the questions come later, right?
Yeah, thank you so much, Jairo. There's I've got quite a few questions of my own, but other people in the chat have written through some really excellent questions, so we'll come to them a little bit later after Felipe gives his talk. Thanks so much, Jairo. That was really, really interesting. What an amazing species.
My pleasure. Thank you all for listening.
Hi. Well, very nice talk, Jairo, and very interesting finding species and very interesting papers too. Very delightful to hear the talk. Well, now I am going to present my talk. Let me give me a minute so I can share my presentation. So, can you see the presentation? Great. Good, good, good. Okay. Well, so as you have already said, I work in Argentina in the National Scientific Technical Research Council and in the National University of Salta, and I am also a member of the Killifish Foundation, which is devoted to the research and conservation of killifishes. I'm going to talk today about 2 species that we described last year. So I'm gonna try to be brief so we can go through the 2 species. So I will go on the generalities of those species, and if you have any questions or you want to go deeper in anything, you can ask later about anything you want to know in more detail. So the first species that I'm going to present to you is Argolebias adrianae. This is small and very nice killifish. This species, we discovered this species near the Iguazu Falls in northeast Argentina. This species lives in temporary wetlands in the middle of the jungle that completely dry up in a moment of the year. This is the temporary wetland. We found this pond next to one of the paths inside the national park. when we were working there with a couple of friends that we were doing a fish survey, and we discovered this small pond. And when we tried to collect here, we collected this new species. It was the first time we tried to collect a fish in the park, in that sampling, and the first net we put in the water came out with a new species, so it was great. And this pond, you can see the same pond here. When it's dry, it dries up several times per year. And so you can see this is the same pond. It's very small when it is with water and when it is dry. So seasonal killifish are a group of fishes that inhabits in Africa and South America. There's one species in Mexico, in Central America, and they live in ephemeral temporary wetlands that dry up completely. They have drought resistant eggs that can endure these dry conditions. The adults die, but the eggs survive the dry season, and then they hatch when the rains fill the pond again. And the most striking thing about this species is that most seasonal killifish species that we know, they live in habitats that have a more or less predictable dry and wet season. Like you generally know that, for example, in summer or winter, you have the wet season, and then you have a period of a dry period, and so on. But the striking thing in this habitat is that there's no pattern there's no predictable pattern of filling and drying. You can go in any time of the year and the pond can be dry or wet. So this imposes, in biological terms, a big challenge for this species, because most seasonal killifish have their life cycle suited to cope with a more or less predictable environment. But living in a very unpredictable environment is an extra challenge for those fishes. Here you have a graph showing differences in the rain. You have the maximum in red and the minimum in yellow of temperatures through the years from December of 2016 to November 2020, and then in light blue you have the rains of this period, and then in dark blue you have the depth of the pond. So as you can see, the depth of the pond, when it's zero, it's dry, completely dry, and this pond in this period dry up a lot of times, and there's no clear pattern of filling and drying. From the point of view of the evolution of this group of species, this new species that we call Argolebias adrianae, here it is closely related to another species that we described in 2023, that is Argolebias guarani, which is very, very colorful species. And it is close geographically, but it is in the Middle Paraná Basin, adjacent to this species that is from the Iguazu Basin. And then we have Argolebias nigripinis, that is another species from the southern portion of La Plata Basin. So we built this phylogenetic tree based on morphological and molecular data of those species. Here you have a drawing of these species. In the left is a male, and in the right is a female. You can see those species have a great sexual dimorphism, and that is very common among killfishes. Males are generally very, very colorful, and females are more brown and a bit more dull. This species was dedicated to Adriana Almiron. She is an Argentine ichthyologist that works with fishes, and we want to acknowledge her work, and by doing so, we want to acknowledge the work of females, of women in science, and to advocate for a science with no gender bias and a more equal and science. So this is a map of this region of South America. You can see Paraguay, here is Brazil, this is Argentina, and we can see the Paraná River here, this is the Uruguay River, and this is the Iguazu River Basin, where we find Argolebias adrianae. As I told you, nearby is the Argolebias guarani, which is below the Iguazu Falls. Argolebias adrianae is up the Iguazu Falls, and the separation of these species, according to our phylogeny, is more or less in the same amount of time that the Iguazu Falls appeared. So probably they were separated by that geological event. And in the Iguazu Fall, in the Iguazu Basin, we also have another 2 species of rebulid killiefishes. One is Garcialebias araucarianus, and the other one is Acrolebias carvalhoi. Well, so this is the same pond of these species in different stages of the drying and filling of the pond. So this species doesn't live all the time under the soil like the species of Jairo, but they spend a part of their lives in the soil too. Well, how we can tell this species apart from other species, among other features, mainly by some coloration characters, like the anterior portion of the dorsal fin with a big black blotch, or the black markings in the females, or some iridescent spots in the pectoral fins, and also because of some osteological and other features. These species present very interesting displays, aggressive displays between males. They are very territorial. They fight each other, making very strong and aggressive displays, and that is why one of the reasons that this species is very colorful. Here we can see the variability of the color patterns of females in the left and the males in the right, and the ontogenetic changes that undergo when during the development of these species. You can see that the older males have a lot of iridescent markings on their bodies. And here is a comparison between other species of Argolebias in the ontogenetic changes of their coloration. And a very interesting thing that we find in this species from northeast Argentina, that are Argolebias adrianae and Argolebias guarani, is the structure of the surface of the egg. If we see the structure of the surface of the egg, of the chorion, we see these ornamentations that we see here, and in Argolebias nigripinis from the southern portion of La Plata Basin, these species present these hairy-like structures that are absent in Adrianae or Guarani. Okay. Well, so we have to talk about another species. So I will want more deeper in Argolebias adrianae. Now, the second species that we are talking today is Hoplisoma osvaldoi. This we described this species last year too. Is the fish in this picture, and this species is endemic from the northwestern portion of the La Plata Basin. This is the La Plata Basin, and in the northwestern portion here, in the Andean region, we have a region with a lot of endemic species, and we have been describing many species from this area for the last 20 years or so. and here we discovered this new species too. This is the Blanco River. It's an affluent of the Bermejo River, and in this river we discovered this new species in small creeks like this one in the picture here. With the habitat of this species has clear waters and sometimes fast flowing waters, especially in summer when the rains is the rainy season, and we have very fast flowing waters. This species has the habitat of this species has a lot of filamentous algae like this one in this picture. And with respect to the phylogenetic relationships of this species, we recovered when we did a phylogeny of the Calictidae family with emphasis on Corydoradinae, using molecular markers and morphological data. And we recovered the new species here in what we called an Hoplisoma palliatum clade. The genus Hoplisoma, you probably know that recently the genus Corydoras was split in many genera, and one of those genera is Hoplisoma, but in our phylogenetic reconstruction, Hoplisoma was not found monophyletic. Here is the other part of the tree that we recover, and you see we have an Hoplisoma sensu stricto here, and another Hoplisoma clade here separated from that one. And also here are several species that were previously in Hoplisoma that are found in another clade. So these three species here are from northwestern Argentina too. So we describe this group as a new genus that is Urcumayu, that means mountain river in Quechua. And we described this new species of Hoplisoma osvaldoi, which is in the Hoplisoma palliatum clade. As you see here, probably in the future, we will have to create a new genus for this group of species. So this Hoplisoma palliatum clade has also two different groups of the species, one northern clade and one southern clade. Here we see the distribution of this Hoplisoma palliatum clade and also of the Urcumayus species. Urcumayus species are in this region, and Hoplisoma osvaldoi is the white star here in the middle of that. And Urcumayus species here are the sister species of Hoplisoma flaviolum here from the upper Paraná basin. And all this is the southern clade of the Hoplisoma palliatum clade, and this is the northern clade of the species. So the new species of Hoplisoma osvaldoi is the sister species of Hoplisoma palliatum. Hoplisoma palliatum was originally Corydoras palliatus, that you probably know because it's one of the most common aquarium species in the world, and one of the first that was bred in aquarium and so on. It was discovered by Darwin in Uruguay, from southern Uruguay, and described later by Jennings. So if we compare Hoplisoma palliatum with Hoplisoma osvaldoi, here is a picture of Hoplisoma palliatum from the type locality here below, the habitat where it lives, and at the right, we can see Hoplisoma osvaldoi and the habitat where it lives. You see, the habitats are very different, and the habitat of osvaldoi is very fast flowing and has a lot of stones in the bottom. And that is related with the morphology of those species. You can see Hoplisoma palliatum is a high-bodied species, and Hoplisoma osvaldoi is more elongated. It has very short fins, like the dorsal and pectoral fins are more shorter, and the spines of those fins are shorter too. And it has a shorter caudal peduncle and more deep caudal peduncle. Those adaptations are adaptations to to habitats of high flow speed. We also studied the development, the ontogenetic development of this species and its coloration and its osteology. We find some also differences in the osteology. And these are the species that I told you before, the Urkumayu species. These species are very, very adapted to those mountain rivers. You can see those are even more elongated than Hoplisoma osvaldoi. And it's interesting, as I told you, because those form a very distinct group, separated from all other species, except from Flaviolum from the Upper Paraná basin. So the interesting thing here is that Hoplisoma osvaldoi here lives together with Urkumayu micracanthus here in the right. As you can see, those species share a very similar coloration, but as I showed you before, they are in very distant places of the phylogenetic tree. And this is because those species are Mullerian comimics. What does that mean? It means that, well, those species, Corydoradine, have venom glands associated with the pectoral fins. And many species show aposematic colorations that enables the predators to easily detect and recognize those species and then avoid them. So many species mimic each other in the same place, so the predators only have to learn one color pattern. So that was previously described for several comimetic pairs of Corydoradine, previously in a paper of Nature in 2011. And well, we find this the same thing here with those two species. Here, for example, you have another Urkumayu species. You can see this species is more closely related to Urkumayu micracanthus, but the coloration is very different. And Hoplisoma osvaldoi is more similar in coloration to micracanthus than micracanthus to Gladice here. This is, by the way, the Corydoradine of the highest altitude in the world. Lives in this river here. You see it's very fast flowing, and it is at 2,500 meters above sea level. We also described the swimming behavior of those species and compared it to species from the lowlands. And we also find several things that I'm not going to go into much detail because we don't have too much time, but they also have adaptations in their swimming behavior that help them to cope with those fast-flowing waters, and that are very interesting too. So finally, I'm going to close the presentation with explaining why is it possible that in this region, in the northwestern portion of La Plata Basin, we have so much endemic species. So this is a topographic map of South America, and we have places. This is the lower portions of South America, and then we have some places that are more elevated. The more elevated places are also the places that have more fish diversity. Why is that? Mainly because the lowlands were under the sea when the sea rises in different periods when the sea level rises. Those portions of South America were under the sea. So those places were clean of freshwater fauna, but the older portions have more time to evolve, and there were more geological and historical processes there that generated diversity. Also, another important thing here, is that there's an ecological barrier between the Andean portion and the floodplain, because those are very different in ecological terms. So generally, the species that are in the Andean portion and the species that are in the plain do not cross from one region to other. Some species do, but most don't. And why is that? That is because those environments are very different ecologically. This is the Bermejo Basin in the Andean portion. You can see it has clear water and sandy bottom with a lot of rocks. And if we go to when this river gets into in the floodplain outside the Andes, it's a more wide river with a slower flowing. It has muddy bottom. There are no rocks at all, and it's very turbid, very murky. So those ecological differences between those areas are preventing most species to disperse from one region to other. And that is the reason that we have a lot of endemic species here in the northwestern portion in the Andes region. And but that makes also another thing is that we have those endemic species have very restricted distributions, and that together with the anthropic activities that we have in these areas, like mining, agricultural expansion, the cities that throw sewage, and we have industries and so on. All that impacts in the aquatic fauna in this region, and because we have species with restricted distributions, many of those species that I was talking before are in danger, in different categories of endanger. So to close this talk, I want to say that that it's important we raise awareness of the fragility of those ecosystems and the fauna that inhabits in them, and we try to make them to conserve them and to make them know. So I want to especially thank this initiative of this webinar, because I think it's very important to achieve that goal. So thank you very much for your attention, and I am ready to answer questions at the end of the webinar.
Thanks so much. Excuse me. Losing my voice. Thanks so much, Felipe. That was fantastic. I really enjoyed those slides at the end showing the topography of South America. I didn't know that. That's really, really interesting. So thank you for sharing that. Valeria and Rupert, are you guys ready to share your presentation? Then we can jump into a Q&A.
Okay, I will share my presentation. Are you seeing the presentation? Yes?
Yes. You can make the slides in full screen, I think.
Okay. Okay. Good evening. Here is 1 p.m. And now I will talking about the two species that we described, two species of Melopus that we described in the sorry, my English is very bad. Our work was entitled Integrative Taxonomy of the Black-Barred Disc Paculs, with the head description of Melopus schomburgki and the description of two new species after named Melopus island and Melopus sauron. This this work was carried out by me and Vitória Pereira, Rafaela Ota, Rupert Collins, Marcelo Andrade, James Garcia, Michel Gegui, Izeni Farias, and Thomas Herbert. And this work was published in the Neotropical Ichthyology in the last year. Miloplis schomburgki always has always been considered a species of easy morphological identification because this species has an vertical black bar in the middle of the body, and it is a principal characteristic of this species. And here we can see this black vertical bar in the picture of the left picture. We can see a female specimen when the black bar is very apparent. And the right picture, we can see a male specimen. Both pictures are from Miloplis schomburgki. These characteristics inspired the vernacular names like black-barreled disc pacu, black-barreled mileus, black-barreled silver dollar, and black-banded pacu. In Brazil, we call this fish pacu cadete. Rupert, can you
Wow. Can you put the I think the slides are going a bit slowly. Can you put it in presenter mode? So I think you're one slide ahead here.
All this time I've been only seeing the first, like the title one.
Yeah, I think we need to click slideshow or apresentação the slides. Excuse my pronunciation.
You can see?
Yeah, if you're able to, sorry, I'm losing my voice. If you're able to start the slideshow by clicking at the top on the presentation, and then you should be able to start the slideshow and it will big screen it. Alternatively, I could get your presentation up from this side. Maybe that's easier. Oh,
Great.
yeah, that looks perfect.
That's better.
Thank you.
Okay. You can see the next slide, number 2.
No, I can't pass the slide.
Oh, is it not
Not
is it
possible
not
to pass.
changing? Maybe I could do it from this side.
Yeah, that might be easier.
Yeah, let's go.
It seems to be lagging a bit there.
How's that?
Yep. Okay. Number 2.
Yeah.
Yes. Okay, here we go. Right. So this is an interesting fish because it's kind of the exact opposite of what Jairo and Felipe were talking about with their fish discoveries. This fish has been hiding in plain sight for hundreds of years, because these are really common food fish in the Amazon. Lots of people eat these. They're really well known in the aquarium hobby. but traditionally they've only been known under one name because they have this this really distinctive black bar, which none of the other members of the group has. They're just so easy to identify, and people have just, always just treated it as Mylopus schomburgkii and never thought any further about it until we started investigating it in a bit more detail. Next slide. So since it was described in 1841 by Jardine based on the collections of Schomburg, and unfortunately the type specimen was lost. So we had to designate a new type specimen, a neotype, for the species. And as you can see, this is the illustrations are absolutely fantastic that that Schomburg did, and they did such a great job of illustrating the fish that he collected in the Rio Negro, where it's from. And you can see there's absolutely no doubt that this is the same group of species that were identified as Schomburgkii back then. But so where we came in was when we did a really big molecular study. Well, Val mainly did all the hard work in assembling the absolutely enormous data set of over 1,000 pacus and piranhas across the Amazon. And we found out that there's the molecular data, the CO1 barcodes were suggesting there were many species hidden in this really well-known taxon that everyone was familiar with. You see this is on the left down here, there's a figure from Val's paper in 2018 from her PhD work. And you see there's at least three, well, it has four clades of Schomburgkii there. And it was clear that there was a lot of genetic structure in this group, and other groups have also come to the same conclusion based on their molecular data. So there was a lot of work to do. Okay, next slide. Val, do you want to describe the sampling you did across the Amazon?
Yes. It was this map is the localities, the points of the localities where I collected the species of the family Serrasalmidae, and I collected in almost all points, but some samples other researchers and colleagues collected for us. So, but the description of the two new species was possible due to widespread sampling of Serrasalmidae over many years and many trips, which resulted in a molecular dataset of over 1,000 species. And
how many trips was this, do you think? There was a lot of field work.
how many? A lot of, but I don't know. I think 20, 25. I don't know.
Oh, yeah, you've seen almost all of the Amazon now.
Almost all Amazonia. And I collect most of the samples, but some were collected, but other research and collects. And the objective, sorry. I can pass the next slide,
Yeah,
please.
next slide.
Okay, yeah, okay. And then the objectives of our study was to delimit the lineages within the Mylopus schomburgkii complex based on DNA barcodes of the cytochrome oxidase gene to had described Meloplus schomburgki and described 2 new species previously misidentified as Meloplus schomburgki. So, based on the results of my PhD work, we increased our sample size to include new locations because we had a small number of individuals of Meloplus schomburgki. We rerun the species delimitation analysis to identify lineages within the Meloplus schomburgki and performed morphological and osteological analysis to confirm or refute our molecular results. So we had described Meloplus schomburgki and described the 2 new species. Next, please. Okay. And we use molecular and morphological data to analyze the Meloplus schomburgki. So what is integrative taxonomy? The integrative taxonomy aims to use multiple lines of evidence to delimit species like morphology, genetics, ecological and behavior data. And the genetic data help us break up difficult crypt species group. So our results show us the divergence in molecular data between the 3 lineages. Meloplus schomburgki was different in the cell CO1 gene from Meloplus island by 7.9%. Meloplus schomburgki was different from Meloplus sauron by 9.7%. And Meloplus island and Meloplus sauron are different by 11%. Hooper, please.
Yeah, so I think Jairo was talking about the divergences in the CO1 barcoding gene that you would typically expect with a different species. Like you said, around sort of 2 or 3%, and you start increasing the likelihood that something is a different species. But when you get to sort of 8, 9, 11, 10%, then you'll be very certain that something is probably likely to be different. You can see on the tree on the right, the 3 main clades of the Schomburgkii, sauron, and Islan are not even monophyletic in the tree, and this is because how divergent they are. And the CO1 gene is not good for making phylogenies, so we're not really confident that they are sort of unrelated to each other in that kind of way. But it does tell you that they're so different, and yet morphologically actually quite similar. So this is kind of an example of a cryptic species. The true cryptic species are the ones where it's really impossible morphologically to tell them apart. But this is more what we kind of call a pseudo-cryptic species, where it kind of been overlooked for many years. But when you look at the molecular data and it divides it into lots of groups, but then you can see those groups, and then you look at the morphology of them, and then you can actually find characters that do support those different groups or refute them in some cases. But this is a kind of a pseudo-cryptic species group. But the morphological data was quite quite small data compared to the molecular. The morphological differences are quite small. So kind of slight differences in the number of vertebrae, for instance. Typically, Meloplus sauron has fewer vertebrae than the Schomburgkii and Islan, and dorsal fin rays. So these are sort of counting, and these are called meristic. These are the sort of counted features of the fish, and they're often very typically used in taxonomy. And so the sauron again has fewer fewer branch rays than the other two. But generally, they are very similar morphologically. Good. Do the next slide, please.
To corroborate our results with molecular and morphological data, we can see some difference in the shape of the vertical bar, dark bar. For example, Meloplus schomburgki has a vertical dark bar with uniform width when compared to the two new species. Meloplus island, in Meloplus island, the vertical bar has a wider region near lateral line, and the extremities do not taper. And in Meloplus sauron, the vertical black bar narrows toward the extremity, forming tapered distal tips. So we can see, I think, you can see the difference in the shape of the black bar in these three pictures. The left, Meloplus schomburgki, in the middle, Meloplus island, and Meloplus sauron in the right. Next.
Yeah, so it's quite, these are nice examples, right? But wasn't always so obvious in the field, telling them apart. But the island seems to, it's more like a sort of chevron shape. Once you get your eye in and you can see them, it's a lot easier. But until you can kind of understand the differences, it's it's quite tricky. The Sauron has a much thinner stripe, so that one's a little bit easier to distinguish, and it's also restricted to the Xingu drainage, so it's a bit more obvious that this one was different. But the other two, Island and schomburgki, are very similar. It takes a little bit of time to get your eye in. Next one, please. So you can see a really nice example here when these two were the fish on the right. These were both collected in the same net, I think. Where were these from, Val? Do you remember? Rio Branco.
Yes, in Branco River, and the same place, the same region, and the same gillnet. We get these two exemplars, but we ate. Okay.
Yeah, so this is a problem with sampling. When these are very common food fish, and when you're in the field, there's quite often you'll fish you're not collecting for the for science. You're putting to the pot to have for dinner, and this was an occasion where these lovely specimens, and because we collected so many of them, these actually got eaten, unfortunately. But it does illustrates the differences between the two really nicely. So the etymology of this species, Island, it's very sad actually. This was unfortunately one of our co-authors lost his son prematurely, so he's kind of dedicated this species to his son's memory.
Yes.
Okay, next one.
And the Meloplus sauron alludes to the eye of Sauron from the Lord of the Rings, when the elliptical body of the Meloplus sauron, marked with a vertical black bar, tapering toward both ends, resembles the famous vertical pupillate eye from the novel. This name was Victoria's idea, because she loves this novel, and she saw the apparent semblance. I don't know who put it in.
Yeah, the similarities were. It was. I think it was a no-brainer that it got called Sauron. It was so just the shape and the color and the eye. So it was absolutely perfect. So yeah, there was no no doubt that was what it was going to be called. Okay. Next slide. Yeah.
And after the publication of the work, we have had some posts like this. So many posts after the publication of the work.
Yeah, it was really nice. It got such good attention on social media that people were kind of appreciating the value of taxonomy and discovering new species and kind of thinking a bit more about a place in the world. Okay.
About the distribution of the 3 species, Meloplus schomburgki is wide distributed in Amazon basin. This species occurs in the Guyana and Brazilian shield. Meloplus island is restricted to the western of the basin, and Meloplus sauron is endemic to the Xingu River.
Yeah,
okay.
I was going to say that they're all sauron is a clear water species from the Xingu, right? So if anyone knows anything about the kind of the sort of 3 main water types in the Amazon, you've got the clear water, which is as it's described, it's very usually crystal clear in a lot of rivers, or just slightly turbid. Then you've got black water, which is like kind of black coffee, black tea, kind of really dark stained water. And then you have white water, which is not just fast flowing water. It's actually like sort of your more sort of milky tea, milky coffee kind of color. So I think these were Island and Schomburgki are both black water. Is that right, Val?
Meloplus schomburgki and Meloplus island too in black water, but not in white water.
Yeah, never white water, but I think also clear water too, right?
Yeah. And we can see that the Meloplus schomburgki, Meloplus island and Meloplus sauron are sympatric with Meloplus schomburgki. So Meloplus schomburgki Meloplus sauron is only in Xingu River, and Meloplus island occur in some rivers in the west of the Amazon basin.
Next one, please.
Here is some pictures of the habitats of Meloplus schomburgki and Meloplus sauron. Both pictures are from the Xingu River. The 3 species feed mainly on aquatic plants, but aquatic insects are also part of the diet of the species. And the 3 species are exploited by ornamental fish marketing too.
Yeah, I should mention. So I think you mentioned at the beginning, but these are the pacu group is very closely related to the piranhas or the piranhas. So they share the same kind of a lot of the same morphology. The main difference is the teeth. So obviously, piranhas have this sort of very, very sharp biting teeth for predating on fins and fish. But the pacus have very human-looking dentition actually, and they have this sort of cutting blades at the front, but also molars at the back for grinding down seeds, grinding up aquatic plants. And I think they spend a lot of time when the forest floods, moving into the flooded forest areas and eating a lot of sort of vegetation and also insects. They're more on the sort of herbivorous side, but they're people call them vegetarian piranhas, but I think the term piranha really should be restricted to the meat-eating Serrasalmus mainly, but people like to call them vegetarian piranhas, but they're kind of technically not really. But they're super nice, super nice fish to specifically to look at in the aquarium as well. And they're really popular. I've seen all three species actually in aquarium shops in the UK, so they're quite commonly exported. Which brings us to the next slide. So this is something I noticed. So this is a fish, the one on the left with the big sort of diffuse blob. This is a fish that's been popping up in the aquarium trade. I think it's come from Peru. They call this one Myloplus blackberry. And we couldn't get any samples of this for the paper because they're kind of rare, but we definitely want to be able to get up and try and do some field work in Peru or get some samples from some colleagues in Peru and see where that one kind of falls out and exactly whether it might be a new species or whether it's just a sort of color morph of eyeland. It could be either. So yeah, it'd be really exciting to understand that one a bit more. And there's another fish on the right here. This was included in the paper. This was a super interesting one. This was called the Tellers Perez fish. Oh, slides have gone. Hang on.
Okay, in the future studies, we intend to analyze Myloplus schomburgki from the Brazilian shield. Since in the Tapajós River basin, we found a lineage with low molecular distance. And this picture A and B, low molecular distance, but with a vertical bar pattern very different from that found in another part of the distribution of these species. So these exemplars, A and B are Myloplus schomburgki. In the molecular results, we had about 1% of difference, I think, in the molecular, but the vertical black bar is not a bar. This pattern, but we can't found another characteristic that we can describe it. Another species, another species for the Tapajós River. So we can we decide to call this lineage Myloplus schomburgki 2.
Yeah, so this was a bit of a sort of not disagreement, but there was a lot of discussion amongst us about whether to describe this one because there was some people who thought this is morphologically very distinct and should be described as a new species because it's so different from the others. But it was actually nested within, with the molecular data, it was nested within the Schomburgkia as Val said, and there was also a difference between the Brazilian shield and the Guyana shield groups as well. So they were separated by the main Amazon River running between them, and they were separated with a very, very small molecular divergence. So in the end, we decided that it's probably better to be conservative and group everything as one species, and then wait for more data and more samples, and also perform genomic analysis where we can do, get much bigger data set of DNA and actually try and work out exactly the relationships with them, because the CO1 data wasn't sufficient to differentiate them. But this is kind of one of the nice examples of working with geneticists, working with morphologists, and discussing the how these kind of traits can appear on the tree and how they can appear in the sample of individuals we got, and how best to treat that taxonomically. So there's never quite often a black and white answer. You kind of have to work with the information you've got and do the best you can. So yeah, thank you.
Brilliant. Thank you so much, Rupert and Val. And yeah, Val, I know you said earlier that you were a little bit nervous about the language, but I think you absolutely smashed it. So thanks so much. I really appreciate you. Really appreciate you presenting your work. It was fantastic to see. Okay, so there have been, as expected, quite a few excellent questions that have come in. I'm very selfishly going to ask one for myself first, but hopefully it will resonate with other people first. Val and Rupert, it's about your talk and about your naming of Malopus sauron, which is a common name, could be Lord Sauron's Pacu, is what we've included it as in the new species report. I just wanted to get your insight into how naming a fish species after something that's popular in popular culture, how that can help generate interest, because I know there's been a lot of media interest about Malopus sauron.
Hopefully.
Yeah, I can try and I think sometimes as scientists you kind of want to be a bit more sort of a bit more low key and try and just sort of get on with the work and be not try and hype things up beyond what you think, though, because the fish is like, it's a nice description, but like in terms of like sort of the interest of the anatomy and things, like compared to Jairo's fish, it was nowhere close to the actual the level of scholarship that went into that kind of discovering these things. So it feels weird in a way having something that you put a lot of work into being kind of getting out there really popular. But as Michael says, it's just so important for people to really realize that we are still discovering new species, and it's not a rare or unique thing. It's just something that has to happen for us to understand the world and how we and how we're affecting it and what we need to do to stop that. It's just it's so important. So anything we can do really to make it you kind of just got to bite the sort of kind of accept that that's kind of the what you have to do at the moment to try and sort of just do anything to raise the awareness of these things. But it does feel a bit strange to because these there were hundreds and hundreds of like 600-700, I think, Michael, is that right, described? Maybe 800 last year, and this is just one of them. So just trying to
freshwater fish, there were 260
out of all fish in total. I think it was probably
all fish in total. Yeah.
So,
I can't remember the number, but a lot.
Yeah, so it's just one out of those, and it got a lot of press, which was amazing. So we're really happy with that. Yeah,
definitely works as a tactic. So Jairo, Juan Pablo Cafrune, I'm so sorry if I've pronounced that incorrectly, has asked, Professor Jairo, when you mentioned that both genera aren't considered true eels due to being phylogenetically away from anguilliforms, does that mean that they don't share a common ancestor with the latter that presents a similar morphology? And if that's the case, would it be a case of convergent evolution?
Thank you, Juan Pablo, for your question. Yeah, so basically what I mean indeed is that they're not true eels in the sense that they're not members of that order. They do have similar body shapes, and that similarity in body shape could be precisely a result of convergent evolution, meaning they're like lineages that are distantly related that evolved that phenotype from, like perhaps living in similar environments, but not because they inherited that from a common ancestor, immediately recent common ancestor.
Okay, brilliant. And there's another question. You've really kind of piqued people's interest in such a sort of unusual fish that it's really sort of piqued people's interest. But Anthony Mazarol asks a very good question. What led the group to even search for these fishes in the mud in the first place?
Well, the group, meaning like, I guess us who described the fishes, we were not really digging up mud and looking for something there. It was more like excavation work that happened in the premises of the nature reserve. And I'm not quite sure exactly why, if they were like, it was the workers that were like doing some excavation for some, I don't know exactly if it was something related to the infrastructure of the reserve, or they were helping people looking for Sicilians, which is another possibility, because they actually are they in that very same habitat we find other vertebrates that are Sicilians. I don't remember the species now. I think it's Sicilia volcani that lives in that area as well. So I don't know if there were like people looking for Sicilians and they found these other things, or they were just doing unrelated excavation work. So yeah, but we were not really there looking to find something in the mud. It was a discovery that the workers of the reserve basically made.
Brilliant. Okay. Thank you. And finally for now, Alireza Kiabani, again, I hope I pronounced that correctly, asks, what do these species feed on and what is their respiratory system like?
Yeah, that's an interesting question. And well, the thing is that we really don't know pretty much anything about these fishes. Like, this is just the very first step towards kind of like um understanding aspects of the biology of a species is to acknowledge its existence, right? And to describe it in the kind of work that we have been presenting here today. I mean, some species have been known, like what Rupert and Valeria were talking about, but in the case of Ophisternum berlinae, it's like the first time that we even like encountered this fish. So aspects about its biology, its ecology, its reproductive biology, feeding ecology, etc., are really things that we need to start working on, and kind of like, if we want to shed some light on these aspects. So we don't really know anything. We haven't really looked into stomach contents. We have very limited series of specimens. This description was based on 7 in total, and we really haven't performed dissections. I feel like is future work that we can at some point, or people in Costa Rica, biologists in the region can try and collect more specimens and start doing these kind of studies. Something I didn't mention is that one of the main differences, right, like this precaudal vertebral number in Berlinae is related to this most likely idea of having a longer abdominal section, and therefore there has to be some dietary, I guess, like implications of that difference in Berlinae versus the other Ophisternum. So having a longer space, a longer abdominal cavity for a longer digestive tract could certainly suggest some dietary differences, which are not surprising because of this very particular habitat. They could be eating like invertebrates or other things from that soil, but we don't really know what exactly they will be eating. And as to how they breathe, well, they do have like a branchial basket with branchial filaments, with gills basically, but because they don't live in the water, I don't think they are really using the gills for like gas exchange. So I have a feeling that most of this gas exchange would occur through the skin, basically through this. So you see the color is highly, like, it's very pink, and this is the result of the lack of pigmentation and the high vascularization, in that area of the body. So we feel like there must be some gas exchange directly from the skin, because there's basically a very tiny layer of skin, highly vascularized epithelium, and the fact that they live there means that they probably are taking their oxygen through, like, from the air, not from the water.
Brilliant. Thank you so much. And this is a question to everyone, but let's start with you, Felipe. What conservation status do you predict for Felipe, for the Adriana's pearlfish and the Osvaldo's Cory? What conservation do you predict? I imagine for the killifish, because it's in a national park, is it kind of fairly well protected? And following from that, are the species red listed, and how do we make sure that conservation action is in place for the species to safeguard their futures?
Okay, great. Yes, yes, as you said, the Argolebias adrianae is in a national park, so this kind of well protected, although it's a small habitat, and even though we were trying to find more ponds of the species in that area, we weren't able to find it elsewhere yet.
But sorry to interrupt you. I don't know if it's just me. Is it audible for people? It sounds like you're underwater to me, but it might be my computer.
No, it was the same for me as well. I just assumed it was just me as well.
100 people all assuming it's just them.
Now, do you
yeah,
hear
that's
me
a lot
better?
better.
Great.
Yeah. Thank you.
Okay. It was those things. Okay. The thing is that yes, Argolebias adrianae is in that national park, and I am now putting the UCN the IUCN submission of that species. I'm doing it now. I think is I don't recall it. I think it's vulnerable or something like that. It's not critically endangered because it is in within a national park. But the thing is that it's only present in a single pond. So we were trying to locate this species outside the national park or within the national park, but in other ponds. We weren't successful at that, unfortunately. I still think it is possible. The thing is that this province, Misiones, in Argentina, has several reserves and national parks, and so, but it has also a lot of deforestation. So outside the protected areas, it's very hard to find natural habitats, and those ephemeral aquatic habitats are um, very fragile because they can be easily filled with earth or drain, so it's easy to lose them. I also think that in that region there are possibly more new species, and what I am afraid is that maybe there are species that have already been lost, and because the more likely place to find those seasonal killifish now in that region is inside the protected areas. So that's a problem, but we are working hard in that region to try to find more ponds of those species and to try to detect new species also. And in the north western portion of La Plata Basin, when that we were working with those Corridoradine, like Urcumayu and Hoplisoma and so on. I only mentioned those, but there are several endemic species in this region, as I told you. And I have categorized it, most of the endemic species, and many of them are under different threat categories like endangered, critically endangered, like Urcumayus gadice is critically endangered, for example. And yes, the thing here is that we have species with very restricted distributions. Many of them are not present in protected areas, and we have a lot of anthropic impacts from many different sources. So I think the situation here is a bit more complicated, especially for some species. There's one species, for example, that is endemic to the city of Salta. The city of Salta is almost 1 million people. So there's a lot of people throwing waste to the river, and that has a lot of impact in this species, for example. So I'm talking with the authorities here, and I'm trying to establish with them some policies to protect those species. But it's very difficult in terms of the you probably know that our country has a huge debt with the FMA. I think that's how you call that. Well, we have a huge debt. We owe a lot of dollars, and that means that we have to export things to pay the debt. And to do that, we only have primary sources like mining and soybean and all that things. So I think one of the biggest problems with the third world in terms of conservations may be the economic model that we have in the world, where those countries are meant to provide primary sources for the developed countries, and that is imposing a lot of pressure to deforest and do more mining and extract more oil and plant more soybean and all that things. So I think that's a very very big problem for all South America, not only Argentina. And I don't know in the long term what will prevail, if it will prevail if the eager for money of human beings will prevail, or we will be a little bit more rational, and we realize that not everything is money, and we need a place to live, we need water, we need air, we need to conserve and protect our world, because there's no another one. And well, that's I don't know. I would like to be optimistic, but I'm not sure. I'm not sure what is the future.
Thanks so much. And same question to Rupert and Val, if Val's still there. Hopefully she is.
So you think you saw the distribution map of the three species. Now they are very widely distributed across almost the whole Amazon basin. So at the moment we're sort of considering these as being least concern, which is good news. But the Myloplus sauron was restricted to the Xingu River. Now, when you say something is restricted to a river, that makes it sound like it's like a small area. But the Xingu drainage is I think is about the same size as Europe. So it's like absolutely vast area. And as far as we know, it's found quite widely in the Xingu drain in the Xingu basin. So I think that one's also probably least concern. But if you might be aware of the Belamonte Dam that was built a few years ago in the Xingu River, which actually catalyzed the iXingu project from one of the US colleagues initiated to survey the fishes of the Xingu, and that's actually quite a lot of our material. I think maybe even the holotype of Myloplus sauron came from one of their projects. So the building of the dam was directly related to sort of there being increased biodiversity survey work and understanding what's going on there. So yeah, the dam has a much bigger impact on range restricted fishes like sort of small little loricariid catfishes, for example, that are only found around the Big Bend, the Rio Grande, the big rapid section of the Xingu, and they're suffering a lot more. But the Myloplus they tend to range fairly widely, especially they'll migrate in the wet season and move into forest. So they use a much larger area. So they're not going to be so impacted by the dam. But the whole area is being degraded by soy plantations and farming in general in the Mato Grosso state. So yeah, it is. That's probably the more vulnerable of the three species, actually, unfortunately. But they're still it's not currently under any significant threat, which is good news.
Brilliant. That was good to hear. And finally, Jairo, same question for the Berlin's bloodworm eel.
Yeah, Michael. Well, I think in this particular case, due to the highly restricted distribution and the fact that all this area is also surrounded by a lot of anthropic, like habitat, like anthropized, like a lot of agriculture, a lot of like deforestation around that area. It looks like we only have it there, and it's like it could be also like a microendemic. So given that, I feel like at least it should be in one of the threatened categories. I don't know if it would be like endangered or critically endangered. I mean, we would have to look at the criteria of the IUCN and do this assessment like with some, you know, care, and based on these yeah, like on these points. But I have a feeling that it would be certainly threatened once we get to work on that IUCN assessment.
Brilliant, thank you. And that brings us up nicely to the hour. So thank you all so much for dedicating a couple of hours of your time to come on and talk with us. There were some really insightful and fascinating stories there. Really appreciate it. Valeria, Rupert, Jairo, Felipe. So yeah, thanks so much for sharing your fascinating stories about these remarkable species. And thank you everyone who attended as well. Thank you for giving up some of your time to watch this webinar. If you've not read it yet, if you've not read all 4 of the new species reports, I urge you to head over to SHOAL's website and click on the news and stories section, publications. Take a look at the new species report. There's loads of fascinating stories inside those reports. And if anybody thinks of any burning questions that weren't answered just now during the webinar, please email them in to info@shoalconservation.org, and I'll be sure to forward them on to the panelists and get back to you very soon. Remember that the webinar has been recorded, so if at any point you or your friends would like to go and watch it, feel free to head over to the website where it will be on from maybe tonight, maybe tomorrow. And feel free to share over social media with SHOAL tagged so that we can repost. Speaking of social media, it would be really, really wonderful if you could head over to SHOAL's channels. If you're not following already, there's lots of interesting information about SHOAL's partnership, SHOAL's partners, the wider world of freshwater conservation. Lots of interesting news on the socials. So we're on Blue Sky, Threads, LinkedIn, and Facebook and Instagram. And if you've not already signed up for the newsletter, please do that as well. We have a monthly newsletter that comes out. And this is the first in a series of webinars throughout this year and potentially beyond as well, every month or every 2 months. So the next one will focus on killifish conservation, spearheaded by Whipsnade Zoo and the Bristol Zoological Society. I'm sure Felipe will be very interested in hearing about that. And that will take place on the 22nd of May. We'll release more information about that in due course. But for now, make sure it's in your calendars. And that's it. Thank you all so much once again. And I hope you enjoy the rest of your day. See you next time.
Thank you, Michael. Thank you, everyone.
Thank you
Bye-bye.
very much.